Welcome to this explainer. Let's jump right into the hidden, microscopic world of the tiny biological machines that literally power the movement of life itself. If you've ever wondered how your cells actually get things done on a day-to-day basis, well, you are in the exact right place.
Just to give us a quick roadmap for today's explainer, we're going to hit 5 main stops. 1. The engines of life. 2. Meet the biological fleet. 3. Under the hood. 4. Hitting the road. And 5. Small forces, gigantic impact. Let's get to it.
Section 1. The engines of life, powering the living world.
So it's this profound, almost philosophical truth, right? Life implies movement. But the wild part is that almost all forms of movement in the living world, whether it's the contraction of your muscles during a workout or the complex division of your cells, they're all powered by these hidden, ridiculously complex nanomachines we call molecular motors. And this brilliantly illustrates our star of the show. Look at this molecular motor literally gripping a cellular highway, a protein track, and using it to haul massive freight, like that huge cargo vesicle hovering right above it. These are the specific machines powering cell locomotion and driving the fundamental transport network of your entire biology. It's basically a microscopic delivery service.
Section 2. Meet the biological fleet, the vehicles of the cell.
So the biological world basically relies on 3 main superfamilies to get this done. Think of them as different types of vehicles in a fleet. We've got the myosins, which cruise along actin filaments. Then we have the kinesins and the dyneins, and they operate on microtubules. Kinesins generally handle anterograde transport, so moving cargo outward to the edges of the cell, while dyneins handle retrograde transport, bringing things back in toward the center. Each family is perfectly adapted for its specific route and cargo.
Now just look at the staggering complexity of this fleet across different life forms. A simple organism like yeast, it gets by with a pretty modest garage, just 6 kinesins, 5 myosins, and 1 dynein. But mammalian cells? We're talking over 40 kinesins and 40 myosins navigating our cytoplasm. And honestly, these numbers can easily triple when you factor in the post-production modifications the cell makes to these proteins. It's a massive, bustling fleet. And hey, these aren't just theoretical cartoons. We're looking at actual high-resolution electron micrographs of quick-frozen individual motor molecules side by side with their schematics. You can literally see their distinct structures. You've got the globular motor domains that act kind of like little feet and the long stalks that hold the cargo. It is absolutely wild that we can actually image these nanomachines.
Section 3. Under the hood, the mechanochemistry.
Okay, let's open the hood. Breaking down the mechanochemistry, it's essentially a stepwise amplification process. First, chemical energy, our fuel, ATP, binds to the motor. Burning that fuel causes an unbelievably tiny conformational change. We're talking just 0.5 nanometers. But step 3 is where the magic happens. A mechanical amplifier enlarges that tiny shift, which is then translated into a massive physical step along the protein track. It's exactly like a tiny gear shift producing a much larger movement. Now, this gear shift or amplifier looks entirely different depending on the motor you're looking at. In myosin, the amplifier is a rigid alpha helix stabilized by light chains that acts as a swinging lever, super mechanical. But in kinesin, the amplifier is completely different. It's a short, flexible element called a neck linker. Different biological engineering, but the exact same incredible outcome.
And here's the exact chemical cycle mapped out for a kinesin dimer. The whole process relies on this hyper-precise timing of binding and unbinding. You can see how losing a phosphate group uncouples the track binding. This quick interruption basically frees up one of the feet or heads, allowing the motor to step forward and wait for the next cycle. It is a highly coordinated, relentless little engine.
Section 4. Hitting the road, how motors walk.
With the engine running, let's talk about how these motors actually walk. They adapt their styles to their specific jobs, and we divide them into 2 main camps. First up, processive motors. These are the long-haul truckers of the cell, individualists holding onto the track for incredibly long distances without falling off. Then you have non-processive motors. These are the team players. They give a quick, fast pull, detach after a single cycle, and let the next motor take over. Kinesin, by the way, is a fantastic example of a highly processive long hauler.
There's actually been a really fascinating debate among cell biologists regarding exactly how these processive motors walk. Think about your own walking for a second. Do these nanomachines walk hand over hand, where one head swings entirely past the other to take the lead? Or do they move more like an inchworm, dragging themselves asymmetrically with a permanently designated front and back head? When we contrast these visual models, the hand-over-hand movement is heavily favored for motors like kinesin.
Under this model, the free head swings completely past the bound head to a new binding site, burning 1 molecule of ATP per step. Visualizing this microscopic sprint inside your cells, it's just mind-blowing.
But do they all walk in the same direction? Well, most of the fleet treats the microtubule like a one-way street, driving towards what we call the plus end. But biology loves to throw us a curveball, right? We've discovered -end directed motors, like myosin VI, that actually drive in reverse. The exact mechanism for how this directional reversal works, how shifting the motor domain placement changes the polarity, is a huge topic of ongoing scientific research right now. We're still figuring it out.
Section 5. Small forces, gigantic impact. The power of scale.
So let's zoom back out and talk scale. By measuring these motors with incredibly sensitive microdevices, scientists have found that a single motor generates roughly 1 to 10 piconewtons of force. By our everyday macroscopic standards, that number is practically zero. It is an unimaginably small amount of force. To really put that tiny number into perspective, if you wanted to lift a basic, everyday 5-kilogram dumbbell at the gym, you would need 10 trillion of these individual molecular motors working together simultaneously just to pull it off. 10 trillion. But hey, do not underestimate them. In the highly viscous, thick, syrupy environment of the cytoplasm inside a cell, these tiny forces are absolutely gigantic. That tiny amount of force is more than enough for a single kinesin motor to grab onto a transport vesicle many times its own physical size and just haul it through the cell at near maximum speed. And keep in mind, these machines aren't just cellular delivery trucks. As we've learned more, we realize their impact touches literally everything. They are essential for setting up the cell's architecture, making cell division possible, signaling, RNA localization, the whole shebang. And because they are so fundamental, if these motor systems break down, it leads to severe cellular failure, devastating diseases, and even death.
We've mapped out some incredible mechanics today. But remember, many of these motors within that massive biological fleet remain completely uncharacterized by science. We barely know they're there. And that leaves us with a pretty provocative thought to end on. If we are still discovering exactly how these nanomachines operate, what other hidden biological engines are silently walking along the cellular highways inside of you right now? Thanks so much for exploring this explainer with me. I'll see you next time.